Okay, so since it is already 3 o'clock, so I'll get started with the TA session. So, welcome. So, welcome to the week 4 TA session of the course Bioengineering and Interface with Biology and Medicine. The course code for this course is NOC 25 underscore BT05, and I am Arjun. I will be one of the TAs for this course. So week 4. So what I'll be doing over for this TA session is similar to the previous 3 weeks. I'll be going over a general recap of the course material that was taught over in week 4, and after that I will have a short doubt session, and then will be solving some example questions from week 4, after which we'll be going over the answers for week 2 of this assignment questions. So we'll get started with the recap. So this in week 4 of this course, what we generally went through was Mendelian and non-Mendelian genetics. Overall, it was 4 parts of genetics along with the clinician's perspective of point of care diagnostics. So this week, the course instructor, Dr. Sanjeeva Srivastava, started with one second. Yeah. So this week, the course instructor Dr. Sanjeeva Srivastava started talking about genetics, and he started about defining the having a general definition of what a gene and a allele means. So gene is a section of your entire genome which codes for a specific trait or a specific protein, and allele is any alternate form of a specific gene is called as a allele. So, keeping this in mind as the definitions of gene and allele, we went on to look at some more terminologies in the initial lectures, specifically what genotype and phenotype means. So, genotype is like something which is defined as the complete genetic makeup of an organism. Small variations in a gene and combination of alleles which combined together gives you the genotype. And phenotype actually refers to the observable trait, how the genotype comes together to give you an observable trait. So that is the difference between genotype and phenotype of an organism. And we also saw the difference between what is a homozygous and a heterozygous system. So in a homozygous system, the both the alleles are identical to each other. So if we have if we have a chromosome, and if we have different alleles, and if both the alleles are identical to each other, then they are called as homozygous. They can be homozygous dominant or homozygous recessive, or in a heterozygous system, the alleles will be different from each other. They will not be identical from each other. So uh, they will be we'll look at how heterozygous systems give rise to dominant systems or codominant systems. So after that, we looked in detail into Mendelian genetics and how Gregor Mendel, a famous monk slash botanist, did some experiments on pea plants, and found some of the interesting mathematical results based on how dominant or recessive each traits are. And he came up with certain laws of inheritance, mainly the law of segregation as well as the law of independent assortment. So, what is the law of segregation says is that the pairs of alleles are separated when the gametes are formed. So, regardless of how your dominant or recessive genes are arranged, they will be separated when the gametes are formed, and those separately give rise to your final phenotypic or genotypic trait of an organism. And what law of independent assortment says is that if we have multiple traits and the dominant or recessive characteristics of those traits are formed independently of one another, those aren't one trait is not dependent on another different trait. So that is what law of independent assortment means. And we also came in further successive lectures, we also saw how we can sort out this how we can use both of these law of segregation as well as law of independent assortment to solve out problems in genetics by using this specific process is called as Punnett square or using this pedigree analysis. So this is how a general Punnett square looks like. We've also seen a lot of times in these lectures provided by NPTEL. So you take out different traits of an organism and then you arrange them like this and then you cross them over and then you find out how different traits come together to form different genotypic or phenotypic differences. And this is a example of how a pedigree analysis looks, where you can find out what is the probability or what is the chance of someone getting a disease versus someone carrying the disease and not being affected by it, and someone being devoid of any disease or a trait that is being homozygously dominant or something. And we are in further successive lectures we also saw how these kind of problems could be solved by probability as well and not entirely by writing out the Punnett square one by one. And in the last set of lectures, before the clinician's perspective on point of care diagnostics, we saw other cases of non-Mendelian inheritance, where in some cases the inheritance does not follow Mendelian laws, law of segregation and law of independent assortment. Rather, it branches into different types of dominance. One which is called as the incomplete dominance and one which is called a codominance. And in incomplete dominance, we saw the example of a snapdragon flower where the dominant red color uh, flower and recessive white color flower gives rise to a incompletely dominant pink color flower. And even when we further cross these pink color flowers in for the F2 filial generation, then we also try to get some of these red flowers and white flowers and some of these pink flowers as well. And in codominant, in case of codominance, we saw the example of how blood groups work. So blood groups are specific carbohydrate chains on red blood cells, and how we also saw how we could identify this kind of blood type that we have in our body by by taking antibodies against these sugars by incubating them incubating these antibodies against this blood, we will be observing agglutination, and based on what type of agglutination we get against what type of antibody, we will be able to perceive what type of blood group we have. And we also saw that how A and B are dominant antigens in this blood group case, and how if a person has A and B, they can give rise to a blood group called AB, and how this is co-dominant in nature. Whereas if we take O type blood, which doesn't have any polysaccharides, which doesn't have any sugar molecules on the RBCs, it will and when A or B crosses over with O, it will give rise to A or B in return. So these are the examples that we saw over the past few weeks. And yeah, that's all for the recap for this session. So it's a very short recap. So we will have a small doubt session till like 3:10 or 3:15 if we have any like doubts or something, and then we'll move on to problem solving assessment. So in the problem solving assessment, I have taken some problems from the previous iterations of this course, bioengineering, the same course, and we'll be trying to solve some of that problems. And whatever problems we have in this week 4, that will be sort of you can get an idea of how to solve them based on these kind of problems. Yeah. Actually, maybe I'll just go on to solve the problems one by one itself, and then we can have a demo session later as well. So I'll quickly jump into these problems. Yeah. So these kind of problems are very like straightforward problems. So question one is, what could be the possible genotypes in context to blood group if the man and woman have a girl child? So I'll repeat the question once again. So what could be the possible genotypes in context to blood group if the man and woman have a girl child? So the male here has a O positive blood group and the female here has a B positive blood group. So what could be the possible phenotypes? What could be the possible genotypes in context?
The
So
first one.
here, sorry,
The first one.
yeah, IBO and IBIO and IBIB, yeah. So it's kind of it's correct. So IBIO and IOIO. So here for a general this thing, when I upload it to the recording and other people see this recording, I'll just like to make a further clarification of this problem, that to solve this problem you don't need to take the fact that this girl child is not going to help you in solving this problem further. All you need to consider is how these antigens will be, how these genotypes will be formed. So we know that if it's a O positive uh, if it's a O positive gene, you will be having the genotype as IOIO, so you won't be having anything else. So I'll just write it here. Yeah. So, but in the case of a female, if it's a B positive woman, forget about the positive and negative here, because we don't need to consider that in this problem. So, if the female has a blood group, she either can have IBIO or IBIB. Yeah. So if either of them cross over each other, we will be getting B blood group for the child. But what kind of genotype we will be having, that will be the question. So A, so the B blood group could cross over with any of this O blood group, and then we could form IBO, or it could cross over with any of this B types and it could form IBIB. So the answer would be IBIO or IBIB. So I'll go on to the next question. Yeah. So Suraj's grandfather is a fitness freak. He has planted several plants in his garden while he performs his morning yoga. Suraj observed the followings for three different plants in the garden. So plant A, when white color flower producing plant is crossed with black colored flower producing plant, it produces a plant having gray colored flowers. When white colored flower producing plant is crossed with black colored flower producing plant, it produces a plant having only white colored flowers. But in plant C, when white color flower plant is crossed with black colored flowering plant, it produces a plant having black and white spotted flower. So choose the correct statement among the following. So this is also a pretty straightforward question. So plant A, white and black gives rise to gray. So it's kind of a incomplete dominant case of an incomplete dominance, which does not follow the Mendelian inheritance pattern. Plant B, black and white gives rise to white flower. So here you can see that white color is clearly a dominant flower and it follows a Mendelian inheritance pattern, and black color is a recessive trait. And in case of plant C, we have white and black, and in the resultant flower we have both white and black spots. We don't have any gray color spots. So this is kind of a co-dominant system where both black and white are dominant and over each other. So this is also a non-Mendelian inheritance, and the option says that Plan C follows the Mendelian inheritance pattern, which is not the correct case. Plan B does not follow the Mendelian inheritance pattern. This is also not the correct case. Plan A follows the Mendelian inheritance pattern. This is also not the correct case because it's incomplete dominance. But Plan B follows the Mendelian inheritance pattern, which is the correct case because we saw that the white flower is a dominant flower and the black flower is a recessive flower, which in turn gives rise to your white color flower, which is a dominant flower, which is a classic case of your Mendelian inheritance. So I'll move on to the next question here. So one second. Yeah. Consider a dihybrid cross between two trees given below that follow Mendelian inheritance pattern. How much percentage of grey short trees would you expect in F1 and F2 generation respectively? So I'll repeat the question. So consider a dihybrid cross between two trees given below that follow Mendelian inheritance pattern. How much percentage of grey short trees would you expect in F1 and F2 generation respectively? So uh, this is also like very straightforward thing where you can either use probability or you can work out the this thing. So the answer will be 25% and 25%. So one way to solve it using probability is, so you can see here that the gray, which has the capital G small g, it is a dominant trait. So the capital G should be gray and the small g, the double G should be green. So the gray is a dominant one, and we know that in if you cross it in F1 generation, so gray will give rise to like one capital gray and then three greens, so which will be 25%. And if you cross it over further, you will get one fourth into one fourth plus one fourth into one fourth, which will be 25%. It will be two times one two by one two, which will be 25%.
Sir, can you go over the F2 progeny once again?
Okay, so we'll just try to write it down actually. So yeah, so we know that I'll just write it here. G is the gray one, and small g is the small one. So the green one, green short plants. So and same goes for the T as well. The capital T will be your tall plants, and the small t will be your short plants. So when you cross it with each other, what will you get in this thing F1 generation? Okay, like, can you list it out like one by one maybe? Okay, I'll write it out here as well. So with respect to the uh, gray and green plants, you will be getting capital G small g, small g small g, small g small g, and small g small g. Yeah. Same goes for the tall and short plants as well. So capital T small t and small t, it will also be like capital yeah. So the percentage of the uh, gray trees with respect to the green trees. So only this will be gray, and all the others will be green. So your gray to green is 25%. So your F1 generation will be 25%. And now in F2 we have to find whether the percentage of gray short trees will be 25% or 50%. So if you cross all of them, if you try to draw a Punnett square here, so so we'll just see how much percentage of trees will be only gray. So this will be gray and this will be gray. Once again, so once again, I'm getting something wrong here. Okay, it's okay. We'll come back to this problem in a while. But I forgot how I did the calculation first. I will come back to this problem later. Yeah. So yeah. So fourth question is, read the given statements carefully. So statement one says two genes segregate to resultant gametes, one in each gamete. Statement 2 says two alleles of a gene segregate to resultant gametes, both in each gamete. Statement 3 says two alleles of a gene segregate to resultant gametes, one in each gamete. And statement 4 says four genes segregate to resultant gametes, one in each gamete. According to statement, which one corresponds correctly to Mendel's law of segregation? So this is like pretty straightforward question. So the answer is like two alleles segregate to form gametes, one in each gamete. So if you have two alleles and one will go into male and one will come from a female. So the next problem, problem 5. Consider a dihybrid cross between a black-eyed brown-colored fur. Consider a dihybrid cross between a black-eyed brown-colored fur guinea pig with a red-eyed white-colored fur guinea pig, um, producing a black-eyed brown-colored fur guinea pig. What should be the genetic constituent with which the offspring should be mated to perform a back cross? So here I have mentioned what a back cross is. So back cross is when you cross it with a dominant, homozygous dominant parent to generate a hybrid progeny. So in the lectures we saw what a test cross is. So test cross will tell you whether the genotype is like homozygous or heterozygous. So so in the test cross, what you do is you cross it out with a recessive, homozygous recessive organism to produce one half and one half of the recessive and the hybrid genotypes. Here you do a back cross to give out a one half to one half dominant, like dominant but homozygous and heterozygous dominant. And just a clarification, like BR is like a capital BR is like brown. Like this is not separate. Capital B and small r is not like separate. It's just capital B, capital brown, capital BR. And then small b is red, small brown, small BR is like white.
The fourth one, sir.
Yeah, so because it's a dominant thing, so correct. So you have to offspring should be mated with to perform a back cross. You have to choose a dominant tree, a homozygous dominant organism. So the correct answer will be capital B, capital B, capital BR, capital BR. So yeah. So question number 6. Consider a dihybrid cross between black-bodied red-eyed fly and a black-bodied green-eyed fly, producing 4 offsprings as follows. Determine the genotypic constituent of the other parent. So yeah, so we know that it's black and red, and we know this is like black and green eyed, and we know the genotypes of all the offsprings as well. We have to determine the phenotype of the parent in this case. So in this case, the black-bodied red-eyed is not like homozygous; it's heterozygous. But in this case, it's homozygous. So this is also like pretty. So the answer is like one option. Sorry, option three. Capital B, small b, capital R, small r. So when you cross the heterozygous capital B small b with heterozygous capital B small b, you will get equal proportion of dominant as well as recessive traits. And on the other hand, if you cross the heterozygous thing with the sorry ho- homozygous dominant with a heterozygous dominant with a homozygous recessive, you will get equal proportions of heterozygous dominant and homozygous recessive. So the answer is capital B small b, small r small r. So applies the rules of test cross and back cross. So you know that the traits are independent. So you take out the black body and the red-eyed traits separately, and then you try to apply the principles of test cross and back cross to apply the following to get the following traits of the offsprings. So question number 7. Suppose a red color allele is not completely dominant over white color allele, encoding for fur color in guinea pig. Uh, Suppose a red color allele is not completely dominant over the white color allele, encoding for fur color in guinea pig. So it results in a like half red, half white kind of color. If a red color guinea pig is crossed with a white color guinea pig, it produces pink colored guinea pig. Yes. If two such pink colored guinea pig are crossed, what is the expected offspring? So this also it's a very straightforward question. So we saw in the Snapdragon example that in F1 generation all were pink, but if we cross pink with pink, what will be getting? So we saw that the genotypic difference will be 1 is to 2 is to 1 again, but those two alone will be pink, whereas the 1 and the 1 will be red and white. The Snapdragon example. So if a pink crosses with a pink, we'll be getting one red, two pink, and one white. So here we'll be getting one what is this? Sorry, red, one white, and two pink. So one red, one white, and two pink. So this will be the answer. So this just follows the law of incomplete dominance. So you apply the law of incomplete dominance over to the F2 generation. So you will be getting dominant, recessive, as well as the two of the incompletely dominant one. That will be the proportion. So I move on to the next question. So consider a man having 6 digits. He is married to a woman having 5 digits. After genetic counseling, the couple was informed that their child will have 6 digits. What do you feel is the genetic constituent of the man respective to the digits? So here, there, so 6 digits, we know that it's a we came to know that it's a dominant trait in humans. We saw it in the lectures as well. So we are saying they're saying that the man will have 6 digits and the woman is having 5 digits. So if the woman has 5 digits, once again I just pull out the pointer. So if the woman has 5 digits, then the woman should have small d. So both of them should be recessive. And then the man has 6 digits. So we are doubtful whether the man will have capital D small d or capital D capital D. So the couple was informed that their child will have 6 digits. So the offspring is definitely going to have 6 digits. It's either capital D, or capital D, small d. So we have to determine the genetic constituent of the man. What will be this man? So from this we know that so from the woman's data and from the child's data we know that this capital D will not be possible because the woman doesn't have any dominant trait with her. So the capital D is not possible. So the child's phenotype should be capital D small d. And if the child less the if the child is going to get the recessive allele from her mother, if from the woman, then the child should get the dominant allele from the father. And in all the cases the child will have 6 digits. So the in all the cases the child is going to get the um, dominant trait from the father. So the father can only if the father has homozygous dominant allele, then this will be possible. So the answer will be capital D, capital D.
Sir,
Yes.
why won't it be possible if there is only one capital D?
So let's just say if let's just say that let's just work out the case then. So let's just say that the male has capital D small d and the woman has small d small d. So if you cross out, only in half the cases you will be getting a child with 6 digits, and in the rest of the half cases the small d will cross over to the small d, leading to the child having 5 digits. But the question says that the child will have 6 digits. So the case where the uh, child has small d is not possible. So that automatically means that the dad also does not have any recessive allele in his genotype. Is it clear? Is it a bit clear now?
Understood, sir. Yes, sir. Clear, sir.
So I'll move on to the next question if no one has any doubts. So consider a green colored maize plant is crossed with a brown colored maize plant, producing the following offspring. So green color maize plant and brown color maize plant are two different traits. Sorry, two different outcomes of the same trait. Green color is dominant over brown color and follows Mendelian inheritance pattern. Which of the following statement is correct in terms of this context? I think uh, I think there is no picture. I think I forgot to. Uh, put the picture here. Oh, okay. So sorry, the picture got pasted to the next image instead of this one. So we can see here that all of them are green color only. It's like none of the plants are white colored here. But the question says that green color is, and the question also says that green color inheritance pattern is dominant over the brown color flower, and it follows typical Mendelian inheritance pattern. So they are asking what of them, which of the following statements is correct. So we already saw that according to Mendelian inheritance patterns, if the dominant and recessive parents cross over and then they lead to same phenotype, same genotype, then both the parents should be homozygous. Then only they'll and wherein one parent is like homozygous dominant and the other parent is homozygous recessive. So then only they can give rise to all the F1 generation offsprings which are heterozygous dominant. So the answer will be, one second. I think I missed the picture here. I don't know. I'll skip this question for now. Like without the picture of the maize plant, like without the picture, an answer key doesn't fit the picture. So I'll skip this question for now. But do you get the idea, right? So according to Mendelian inheritance, if we have this kind of picture, this kind of offspring, where we have all of them are green, then this option will be correct. Both the parents are homozygous. So in another case where we have equal amount of green and brown color maize plant, then this case will be correct. Where one parent is heterozygous and whereas the other one is homozygous. In that case, it will be an example of a test cross. So I'll move on to the next question because this question is not clear. I'll try to I'll try to find the picture for the question and then when I post it on the Google Drive, then I'll paste the proper picture for this question. So now consider 10th question, like last problem statement for this week 4 problems. So now consider the brown colored maize plant is crossed with another maize plant producing the following offspring. So same, green color is dominant over brown color and follows Mendelian inheritance pattern. So brown color is crossed with another maize plant. So they haven't mentioned the color for this plant. If these maize plants are crossed among themselves, what is the expected genotypic ratio of this offsprings? So that is kind of similar. So it will give you 1:2:1. So yeah. So following mental inheritance, they are asking if the brown color maize plants are crossed among themselves, then what will be the genotypic ratio, which will be 1:2:1. So that's all the problem statements I have for week 4. So I'll leave the room for a short doubt session. Meanwhile, I'll try to solve the unsolved problem in the third or fourth question of the problem statements. I'll leave some time till maybe 3:45 for you to recollect those thoughts. After which we will try. Uh, I'll try explaining the week 2 problems of your assignment problems.
Sir, I had a question, sir.
Yeah,
Sir, about the independent assortment of alleles, sir, we
yeah.
now know that there are linked genes, like linked genes has been discovered, like a few years back, like not a
Yeah.
few years back, like some decades ago. But so now, do you think that these old laws hold true to this day as well? Because uh, the existence of link chains diminishes the law of
Yeah.
segregation, the law of
In
independent assortment. So,
Yeah, in that case, so we also saw like some of the see even the law of dominance, like law of sorry, yeah. So law of inheritances, we saw law of dominance doesn't hold true for almost all the cases. Like we also saw that there are cases of uh, this co-dominance as well as this in- incomplete dominance. So there are deviations from this, but the laws of inheritance just says that it holds true for a majority of cases. And there are other cases as well where you can see that there are no two different states like brown, green or brown or something like that. There will be like n number of varieties, and among them the dominance will also vary among them, and the final trait that is exhibited in an organism is just dependent on the proportion of the dominant allele that is expressed. So those kind of cases also are also there. These are very simple cases where we can see only like uh, two alleles from one allele from father and one allele from mother giving rise to an organism.
okay, sir, understood, sir.
Oh, okay. I got it. I got it nowadays. So actually, I made a small mistake in drawing the Punnett square for the second generation. So what actually happens here is, yeah, okay, I'll just can I use an eraser? Yeah. So while drawing a Punnett square for the second generation, you just take one allele from one would away from each other for crossing over. So one will be capital G and small g. So when these cross over with each other, these won't give you gray plants. This will only give you green. These all will give you only green plants. So out of all the 16 plants, only 4 plants will give you uh, gray trees. And short trees will also follow the same traits. So the total number of uh, once again, I just put out. So the total number of gray short plants will only be 4 out of the total 16, which is like 4 by 16, which is like 25%. That's how we get your answer. While uploading the PPT, I'll try to put out a proper Punnett square for this generation. But this is generally how we solve this problem. We can also take out the case of probability for each of these cases and solve it that way as well. Okay, it's 3:45, so I'll just go on to we'll just discuss the week two assignment session. So week two, as we know, it was on PCR and all those questions. So we'll just quickly go over all those questions and how we uh, go around solving those questions for answers. So these are questions that you guys would have got in your assignment, and these are not like problem statements from previous iterations. These are like your iterations. So um, you guys might have all by this time you guys might have already come across this answers for this one. So we'll just be solving them, seeing to solve them to see how we go around solving them. Once again. Sorry, that was I just got a call. So we'll just start. So Anisha wanted to perform a PCR experiment to amplify her gene of interest. She chose the following two primers. So there is a forward primer and there is a reverse primer. So forward primer from 5' to 3', there is a sequence. And from reverse primer, there is it's a 5' to 3', there is a sequence. So she followed all the steps meticulously but still did not get any product. What could be the possible reason for this? So the answer is pretty straightforward. We have a forward primer 5 into 3 prime and there is a reverse primer 5 into 3 prime. But it will run from 3 prime to 5 prime end. So saying that this 5 prime will go there. So what would happen is if you compare the 3 prime end and the 5 prime end of the reverse primer, both of them will be complementary to each other. So C to G, C to G, T to A, T to A, C to G, T to A, and all those. So it, it's just complementary to the whole sequence. So what will actually happen is, so it will form a primer dimer. What you will observe is you will observe something called as a primer dimer. So primer dimer occurs when primers are too complementary to each other that they bind to themselves and not to the template. So the correct option for this one is the primers are complementary and will form dimers. Yeah. So this will be the correct justification for this. So I'll move on to explain the next question. So a plasmid has multiple restriction sites. However, some of them were blocked due to specific modifications on them. So there are like a number of restriction sites, but some of them are blocked. So digestion using ADE resulted into bands of 6 kb, 8 kb, and 1.5 kb. Digestion using BEA resulted into bands of 8 kb and 7.5 kb. Digestion using BCE resulted into band of 15 kb. Based on the above given information about plasmid map and gel pattern, which of the following sites would be blocked? So normally you can also go around solving this problem like by seeing how if you cut AD and how would be the plasmid pattern, etc., etc. What, but if you ask me logically how I will go around solving this. So the whole plasmid pattern is like 5 plus 1 plus 3 plus 5 plus 1.5, which is 15.5 kb. So uh, 15.5 kb. But you find this one. So 6 plus 8 plus 1.5 is 15.5. 8 plus 7.5 is 15.5. So nothing should be wrong with these restriction sites. But digestion using BCE resulted into band of 15 kb only. So there is something wrong with this restriction site. And if you take the case of E, E is also present in this, and the digestion using E didn't result in any uh, wrong. We resulted in a 15.5 kb. It resulted in fragments which amount to 15.5 kb only. So, based on the given information, only B and C should have been wrong. So the answer should be B or C. B and C. Did you get like? Can you guys get how this thing? You can also go around the long way, solving like cutting down each plasmid and seeing whether it's cutting or not. Whether B, C, and E. Cutting down B, C, and E gives you 3 kilobases and all those 4.5 kilobases and 10 kilobases and etc. etc. But the shorter way to go around this is to perform this kind of uh, analysis and see which ones are not working.
Sir, can you just iterate the long process once, sir?
So the long procedure is simple. So just cut down A, D, and E. Yeah. So I just click laser point. Huh. Sorry. Yeah. So cut down A, D, and E. So if you cut down A, D, and E, what do you get? 5 plus 1, which is 6. 1.5, and then 3 plus 5, which is 8. So A, D, E are working fine. So once again, you work on B, E, and A. Instead of D, you have to put B. Yeah. So in this case, what happens is uh, you have 5 kilo bases, which is not here, but you have 5 plus 1 plus 1.5, which is 7.5, which is here, and then 3 plus 5, which is 8. So, uh, so cutting down A, B, and E should give you 5, sorry, 7.5 and 3 and 5. But instead of that, it's giving you 7.5 and 8. So from this itself, it's pretty obvious that B is not functioning. Do you get it now?
Yeah, sir.
Yeah,
Yes, I
and
got it.
huh. And finally, if instead of A, let's just say that we are cutting down on C. You already know that now B is not working. Instead of A, let's just say you are cutting down C. If you're cutting down C, you should get uh, 2.5, 3, and 10 kb. But you are getting only one band of 15 kb. So E, we already know that it's working well twice. B, you know it's not working. So C also should not be working. That's when only, and you get only one band when you cut only one side. So it should give E is working fine, but B and C are not working. I think the question should have been 15.5 kb, but I don't know if it has 15.5 kb. It would have been less confusing, but I don't know why they gave 15 kb. But that's okay. Like in general, that's how you go around solving this kind of problems. Okay, then. So
Got it,
I'll
sir.
yeah I'll go on to explain discuss the next problem. So I think this problem I already explained in the previous TA session as well, then, but I'll explain it again. So Ramesh has been provided with the map of a plasmid P3000 whose size is 15 kb. So we just draw a plasmid which is like 15 kb. Yeah, 15 kb. He observed the following fragments with different combinations as mentioned below. So there are different restriction enzymes, and upon there are different restriction enzymes, and their combination of usage gives different kinds of fragments. So upon cutting the plasmid with BAM H1 and ECO R1, which are the following fragments which would Ramesh observe? So Uh, we'll first start from the bottom where the question plasmid BAM H1 and ECO R1 are there. So ECO R1 and STU1 give 6 kb and 9 kb. So let's just say this is randomly say that this is ECO R1 and this is STU1. So ECO R1 and STU1 should give you 6 kb and 9 kb. So this should be 6 and this should be 9. So let's go at the next case. BAM H1 and STU1 give 9 kb and 6 kb. So we already have STU1. Instead of eco R1, we should see where BAM H1 will fall. And we know that so the placement of BAM H1 related to this will depend on how the fragments will be distributed. So BAM H1 and STU1 giving 9 kilobase and 6 kilobase. So if you take the usual case where this is 9 kilobase and 6 kilobase, BAM H1 will fall on the exact same place as ECOR1, which is not possible. So we'll take the opposite case where this fragment from STU1 will be 9 kilobases. So if we consider here as sorry BAM H1, BAM H1. So this will be 9 and this will be 6. So based on this, the space between eco R1 and BAM H1 will automatically be 3 kilobases. Yeah. So they are asking upon cutting the plasmid with BAM H1 and eco R1, BAM H1 and eco R1, which are the following fragments would Ramesh observe. So if we cut BAM H1 and eco R1, we'll be getting 3 plus whatever rest of the size is 15 minus 3, which is 12. So we'll be getting 3 kb and 12 kb, which is the answer. Yeah. Sorry, I got a disturbance. So we'll go on to the next question. Yeah. So you have been given a plasmid of 9 kilobases. You have to perform a cloning experiment for which the target gene is GFP. So we have to clone GFP into the given plasmid. Given below is the schematic representation of the plasmid and the target gene. So the target gene is GFP and plasmid is here. For the target gene, we have EcoR1 and NOT1, which is like here. And yeah, the GFP is successfully inserted in the plasmid, which is 0.7 kb. Choose the correct statement. So this is also like a pretty straightforward question. So the answer would be the size of the plasmid after cloning indent would be 8.2 kb because we are cutting EcoR1 and NOT1, which is like 1.5 kb. So it will be this fragment will be like spliced off between NOT1 and EcoR1, which will be so 9 minus 1.5 will be 7.5, and then we are integrating a 0.7 kilo base plasmid, which will be 7.5 plus 0.2, point 0.7, it will be 8.2 kb. So this is also a pretty straightforward question. I'll go on to the next. Yeah. So read the given statements carefully. So lactose binds to the RNA polymerase and increases efficiency. Lactose binds to the repressor protein and alters its conformation to prevent it from binding to the DNA. Lactose binds to the activator protein, which can help the RNA polymerase bind to the promoter and begin transcription. Lactose prevents premature termination of transcription by directly binding to and deforming the DNA. Which of the given statement is correct regarding the activity of lactose in promoting transcription of lacZ? Like this is also a very theory based question. The answer is statement 2, which is lactose is a repressor protein and it uh, alters its conformation from, like to prevent it from binding to the DNA. It's a question based on lack of on. So I'll move on to the next question. So look at the given figure carefully. So there are like one and two. Real-time PCR was performed with a breast cancer and a normal genomic DNA sample from the same patient. So this is the breast cancer DNA sample and this is the normal DNA sample. What would be the most appropriate reason that fluorescence did not increase after cycle 30? So this is a log of fluorescence, and fluorescence kind of saturated after this point, and after cycle 30 both of them saturated. So what could be the reason? So there could be a lot of reasons why PCR signal saturates. One of them will be like primer molecules would have been replaced, or all the template molecules being used up is not a reason, because template based on the denaturation cycles, the templates can be uh, infinitely generated again and again. DNA polymerase becoming limited in the reaction mixture, that could also be a reason, but most probably the answer would be like primer molecules have been placed in the reaction. So usually what we do is polymerase we add more than what we intend to do, because it can generate additional copies than intended as well. So the usual reason is primer molecules that have been placed. So question number 7, read the given statements carefully. Agarose gel acts as a molecular seal which separates nucleic acids based on molecular weight. DNA molecules move towards anode and DNA binding dye is mixed, which fluoresces in UV light. Agarose gel acts as molecular seal which separates nucleic acids based on molecular structure, and DNA molecule move towards cathode. So, which of the following statements, combination of statements, is or are correct about agarose gel electrophoresis? This is also a very straightforward question. So, only A, B, C are right. Like DNA moving towards anode, that we already know, because DNA is a negatively charged molecule and it move towards the anode, which is a positive terminal in the gel electrophoresis setup. And DNA binding dye, usually we use ETBR or Ethereum bromide ETBR, which fluoresces in UV light, which is correct. And agarose gel acts as a molecular seal which separates nucleic acids based on molecular weight. So the smaller molecular weight DNA moves farther, whereas the larger molecular weight moves shorter, like moves less farther from the loading area. So statements A, B, and C are correct. So question number 8. Shruti wanted to perform a PCR reaction. Therefore, she decided to design a primer. She calculated the melting temperature of the designed primer should be 64 degrees Celsius. She selected the temperature flow in thermal cycler as 95 degrees Celsius. 64 degrees Celsius and 70 degrees Celsius for denaturation, annealing, and extension, respectively. Choose the correct statement from the following. So, they are given the melting temperature of the primer to be 64 degrees Celsius, and they are also given the denaturation, annealing, and extension temperature. So, we have to choose the correct statement from the following. So, options are: the PCR reaction would fail as the extension temperature needed to be set at 60 degrees Celsius, which is not quite right because extension at 70 degrees Celsius is correct only. The PCR reaction would fail as the denaturation temperature exceeds the melting temperature of the primer, and the primer would also denature. Uh, could be, but it's not always in the case. The PCR reaction would fail as the annealing temperature was needed to be set at 72 degrees Celsius and extension temperature 64 degrees Celsius, which is quite opposite. Shruti has not done any mistake, and the PCR reaction is most likely going to be successful. So, which is the correct answer, like, because all the steps are like right in this case. She has set the denaturation, annealing, and extension temperature in the proper ranges. Denaturation will be always in 90 to 95 degrees Celsius, since the Taq polymerase can withstand those temperatures, and it will lead to the proper separation of the strands. Annealing will should be at 64 degrees Celsius for effectively binding the primers to the template, and 70 degrees Celsius to be extension, since the polymerase can act in that temperature. So, question number 9: read the given statements carefully. Assertion is that a vector has a streptomycin resistant gene of its own. The Hin3 cut site is present in the penicillin resistant gene. If Hin3 restriction digestion was performed to insert the gene of interest, the bacteria which did not survive in penicillin mixed plate but survived in streptomycin added plate are the required transformants. So, reason is the unsuccessful transformants would have made the bacterial cells sensitive to streptomycin. So, which one of this is correct? So, this is also a pretty straightforward question. But so, like, if the gene is resistant to streptomycin, like, the transformants couldn't have been sensitive to streptomycin. Eh, and contrary, it would have been resistant to streptomycin. So the reason is false, but the assertion is true, because if you are cutting the Hin3 cut side is in penicillin gene, so if you are cutting Hin3, you are cutting the penicillin gene as well. So the penicillin resistance would not have been coded. So the transformers would not have survived the penicillin mixed plate. So, last question. One of the popular methods of transformation of recombinant plasmids to host cells is to perform heat shock transformation. What can be best explained as the role of calcium chloride during heat shock transformation? So, this is also a very theoretical question. So, calcium chloride is like, um, what to say, it kind of gives a positive charge to cell wall due to the uh, presence of calcium ion. When calcium chloride ionizes, it gives to Ca plus, which gives the positive charge to the cell wall. So that's all I have for uh, this thing. The answers you guys would have already known for this questions, so I'm not going to run over them again. So we'll have a down session till the we still have till 5 o'clock for the uh, TA session. So till then I'll keep this room open for doubts. If anything you guys can ask, or I'll see you guys in the next TA session, which will be from 22nd February from 3 p.m. to 5 p.m. Okay, so if there are no doubts, I'll see you guys um, on the next Saturday, which is 22nd February, 3 p.m. to 5 p.m. Thank you.